All right, I'm starting on this lecture. We've already gone through a lot of this, so I'm going to go quickly through the first several slides, and then in 11.3 I will slow down a little. So as we go right here, this is the cell cycle. As we wanted to remind you, there are several parts of the cell cycle. The majority of the time you are in interphase. However, there are periods where we have studied, which is cell division, which happens in the M phase, which is made up of mitosis and cytokinesis. The interphase is made up of three different parts, which has the cell phase, which is cell growth, S phase, which is DNA replication, and the G2 phase, which is preparing for mitosis. All three of these is also where the cell is just doing its normal cell processes. During interphase, as I said, it's 90% of the cell life. In G1, there's growth and normal functions. G2, it's going to be continued growth and preparation for vision. And S is DNA synthesis, which means that we are copying the DNA, a process we will learn about shortly. Mitosis is step one, is where you can actually see the chromosomes. It's visible, and the mitotic spindles begin to form. It looks like this. They're visible, they're in the X form because they've been copied, and here are the spindles forming. The next one is going to be metaphase, where they line up in the middle, and that is so they can divide. Here they are lined up in the middle. These are the chromatids. This is the chromosome in the centromere is the beginning, and these centrioles will connect at what's called the kinetochore to split them apart. At anaphase, you have the sister chromatins are pulled apart, and we have these chromosomes that are moving to both sides of what will be the new cells. Also, don't forget that other organelles are kind of getting divided here. Then we have telophase, where the mitotic spindle disappears. Nuclear envelope begins to form, chromosome coil back to chromatin, and cleavage furrow begins to form. So this is basically getting ready to be a brand new cell. Here you can see the nuclear envelope forming, the spindles going away, the cell starting to pinch in, and we will have two new daughter cells. Cytokinesis is the process of the cytoplasm and organelles getting split up between the two of them. You are forming two brand new cells. The cleavage furrow is happening in plants. There's a cell plate, and then of course the easiest kind of way to see that we do not have chromosomes anymore. They are all done. Here is the way that you get the cleavage furrow in the animal cells. You can tell there's no cell wall. Instead, it just kind of pinches off to make two new cells with two new nuclei. That's why we had to divide earlier. And then we have the cell wall forming here in plant cells. As you guys remember, plant cells have the cell wall, which is important and gives it structure. Now I am going to move on to the cell communication, which you guys read about. So here you have to really think about, how do you think the body repairs itself after injuries such as a cut on a finger? How long do you think this repair process continues, and what do you think stops the process? So normally we would do this as a pair deck, but I'm having you guys do this individually, so I do want you to consider this. How would we know? And really the trick is there's going to be signals, and the biggest signals that we will learn about is this cyclin signaling, which is going to happen between cyclin and CDK. And as a result, you will see that as cyclin increases, it's going to lead to the start of cell division. And then once the cell divides, it drops down again and moves back up again with the enzyme binding to it to make sure that it works. Cyclin binds the enzyme to produce mitosis promoting factor, and then of course if you guys remember, enzymes have special shapes. The special shapes means that this enzyme is now going to be engaged and it's going to help to form again. So the questions are, how long do you think cyclin production would last during a typical cell cycle in embryonic clam cells? During which part of the cell cycle does cyclin production begin, and how quickly is it destroyed? And you guys can see that right here we're looking at about 75 minutes, right, between here and here, and then they continue to do that, and then it's destroyed pretty quickly. As you can see that it breaks down in this green phase. It's getting broken down in about 20 minutes, and then just continues to cycle over and over again. And the cell cycle, cyclin production is produced during interphase. As you guys can see, this would be the interphase part. So I know I'm going quickly through this, but you guys can pause and review through them whenever you want. This is the checkpoint system, which we saw in the Ed puzzle. And a reminder, we have different things that happen at each checkpoint. You have to check to make sure that there's nutrients, that the DNA is not damaged, and that the growth factors are present at the first G point. Now, of course, at the first checkpoint, G1. Now, not all cells always just continue to divide. Some of them go into a resting state, and we talk about how that could be neurons which don't divide, and they stay in a resting state, which makes neurological damage so difficult to recover from. Here is DNA synthesis, and that's where we have to make a copy of the DNA, which of course you must do if you want each cell to have its brand new DNA. And then we have G2, which is one, again, checking to make sure that your DNA synthesis went well and you don't have mistakes in your DNA, because if you do, that means that every single cell from that point on that uses that DNA to replicate will have the same problems, and we're making sure that the cell is ready and has a good size for that. Then we have the metaphase checkpoint, and then we're trying to make sure that the chromosome spindle is attached correctly, because if it's not attached correctly, you could imagine how each cell would not get equal size of DNA. We watched this cell cycle in cancer already. So apoptosis is programmed cell death. This is an important concept. It should be inside your vocabulary if it's not. And cells break apart, and then they're used for their parts. And then the white blood cells will go ahead and identify a damaged cell or a cell that needs to be destroyed. And as a result, the apoptosis begins. It's destroyed by the cell, your own immune system. And this happens all the time. It might sound bad to have programmed cell death, but it's really important. If you did not have programmed cell death, look in between your fingers. There would still be webbing in between them, and your toes or tails would not go away. And then, of course, really important, it's important for removing damaged cells or damaged DNA. It helps prevent cancer. If you could imagine a cell that does not have is not signaled for cell death, that has a problem, would continue to have this unmitigated growth, which leads to cancer, and that is not good if you're trying to live a long time. As you guys can see, here's the cancer. You have abnormal cell division, so you can see that multinucleated cells, there was a mistake there. Multinucleated means that there are too many nuclei, and here you don't want to have two nuclei. That means that they didn't have a proper cell. Here you have multipolar mitosis. So normally when you have two poles and it gets pulled to two sides of the cell, in this one you are having them pulled to three sides, which is going to mean that each cell is getting a weird amount of DNA. Sometimes you can just get stuck in mitosis. It never fully divides. Here you have nuclei that never separated. Here you have weird cell shape. So these are all the things that are being checked for. Lagging chromosomes are showing that they're not pulling apart all the way. And then, of course, you have your normal cells that are in this process. So what you're doing, if you have mutations in DNA or issues in controlling the cell cycle, you will have cells that are not functioning as they are supposed to. One of the most important parts of cell division and controlling cell growth is contact inhibition. This is one of the examples of cell growth that I want you guys to really consider. What they do is, because they are touching each other, it inhibits cell division. So what they are growing in a glass plate, what they are showing is they are removing some of the cells, and then as a result there's a space. Because there's a space, these cells are no longer touching each other, and then they realize that we should grow to fill in that space. That's kind of how healing happens, and they continue to divide until the entire space is filled out. And so here you have normal cells with growth factor in each one, and you guys can see that normal cells, when you add growth factor, they would continue to grow. If you get rid of growth factor, they would not continue to grow. Whereas cancer cells, it doesn't matter if the growth factor is added or not added. What would happen is that they would grow anyway. So what they are doing is they are not receiving the signal appropriately. Just like right here, we would see that cancer cells would just continue to divide without any kind of stopping, because they are not receiving the signal to stop appropriately. That's what happens in tumors. You have benign tumors, which are abnormal growths but not cancerous and do not spread. And you have malignant tumors, which invade and destroy healthy tissue, and they can spread and get into the bloodstream where it metastasizes, and that's really problematic, because once it gets into the bloodstream, it can go to the rest of the body. So cancer can happen as a result of genetic predispositions and also exposure to different chemicals that can increase your risk of cancer, such as UV radiation, chemical exposure, radiation exposure, cigarette smoke, pollution, age, and genetics are some of the other risk factors that can really just lead to it. Once you've continued to copy yourself over and over again, which is age, mistakes are bound to happen. And all these other things can disrupt the functioning of the cell. And as we talk about mutations, inherited BRCA1, there were some pretty famous cases where, say, Angelina Jolie realized that she did a genetic check on herself. She realized she was at high risk for breast cancer, and she had a double mastectomy where she had both breasts removed so that she wouldn't get cancer. Then, of course, sometimes you just get a mutation that leads to cancer. As you look at this, I'll probably have you guys check out one of these TED Talks on your own. I'll put it in the assignment, and I think it's really important that you kind of just understand that, because that's what we study when things go wrong. How do we treat cancer? Surgery. You remove the tumor. You do high energy radiation, which kills cells so that you can find the ones that are rapidly dividing. As we learned, that's why people lose their hair when they do this kind of therapy, because hairs are rapidly dividing cells, even though they aren't cancerous. Chemotherapy, which is intended to stop DNA replication, stop mitosis and cytokinesis, stop blood vessel growth, so that you can basically choke out the cell that has cancer and is dying. So that takes us to the end of this, and I'd like to stop.